So today, it is really my pleasure to introduce Jon Boek, who is a professor in the department of Human Genetics at McGill, as well as the director of bioinformatics at McGill University and Genome Quebec Innovation Center. So Professor Boek leads so many committees and initiatives at McGill, Canada, as well as the international level, really to support, obviously, research in genomics, but also all the infrastructure that's required for this type of work. But today he'll be talking to us about his research in genomics, as well as the McGill initiative in computational medicine. So please welcome Professor Boek.
Thank you. All right, so well, thank you for the invitation. It's a pleasure to be here. As you can see from the picture, I work in the building right next door, so it's very nice actually well, although it doesn't really look like that, but on most days, but it's really, I've done now this in a few other departments, and it's very nice, I think, to have these opportunities to meet and see what others are doing next to us. In choosing what I was going to talk about, I didn't want to go too deep into genomics itself. I thought that it'd be good to give an overview of some of the things that are happening at the Genome Center. So I will talk about some of my research, but I'll also take a sort of a broader perspective. I thought it might be interesting to some of you to know what's happening in the world of genomics these days. So I'll jump right in, and then at the end, I also want to talk about this McGill initiative in computational medicine. That is something that started about 3 years ago that you might have heard about or not, but I thought I'd describe that as well a little bit. So just again, to give a bit of context in terms of where I work and what I do. So the Genome Center next door is one of the centers that's funded as part of Genome Canada. So Genome Canada has been funding genomic projects over the last 20 years now, quite heavily. So they're funding all sorts of different research projects because genomics is useful in context of human health, but also in other areas. And so they're funding projects, but as part of their mandate, they've also been funding platforms that actually support these projects in data generation and data analysis. So two of these platforms are here at McGill. So the sequencing platform and then the center that I lead, which is the Canadian, I'm going to get the name right, Computational Genomics Center. So just going quickly over what's happening at the McGill Genome Center itself first, just again to give context, and then I'll go into much closer to the type of work that I do. So the Sequencing Center has all of the latest tools and toys for sequencing. So that, again, this is offered broadly to all of these projects that are funded in genomics in Canada. So more than half of the sequencing we do is actually not for internal projects, but really for projects across the country. So again, I won't do too much intro, but I wanted to give you an idea of some of the exciting things in terms of new technologies also that we're supporting. So some of so the idea that we can sequence DNA has been around, and we're doing this quite a bit, but some of the exciting developments are now around new sequencing technologies. One of the big change is the fact that the current technologies really allow us to only sequence short DNA fragments, while now there's more and more technologies in production that allow very long sequencing. So there's different applications once we can sequence these very large fragments of DNA using these instruments. There's really some changes in what we can do. It becomes more, for instance, sequencing the human genome is costly, but it's also very difficult if all we have are these very short DNA fragments. It's quite helpful to have these longer DNA fragments to help us assemble those small pieces. So that's one of the development and one of the application. The other exciting development, and I was discussing that actually just now at lunch, is really the single cell technologies. Well, sorry, I'm going ahead of my slides. This is still just talking about the long reads, just showing the fact that there's now new technologies that are coming around, like PacBio and Nanopore, that allow us to sequence a contiguous fragment of 20, 50 kb, in some cases even longer. So that's really sort of a big shift in terms of what we can do. What I was started to describe is this other big direction or new direction for these genomic technologies, which is single cell genomics. This is a platform that's led by Yanis Fragoussis at the center. And really this is applying the same type of sequencing technologies, but where the sample preparation allows us to do single cell, again, at a scale that wasn't possible before. So it used to be that we could do, dozens or less than 100 cells at a time. Now we really have working, I would say, in production experiments where we can do thousands of cells, and that really changes the types of application. So the idea that we can really look at both the DNA and the transcriptome of these cells systematically across thousands of cells, really, I think it's quite exciting application. In terms of the main things we do, these were two examples of sort of upcoming technologies that were integrated into mostly research projects. But there's also a lot of work going back to just regular sequencing, where these platforms are now being used for discovering disease genes and for Mendelian disorder. So this is something that's been so successful in the research context over the last decade, I would say, that the transition is now to really use this in more clinical settings where, so again, it started where, we would try to see if these technologies would work to identify disease gene, to the point where now this is really driven by hospital that want to have a way to sequence individual in a clinical setting such that they can report back on variants. And so that means that some of the technologies that are currently used at the Genome Center will probably be in the hospital in the near future. Similar work for rare Mendelian disease is also applied in cancer. So again, I think pretty exciting to think that what we were doing in the research realm with taking these tumors to look at what type of genetic mutations they have, now the cost makes it such that it becomes appealing to do this really in a clinical setting, to decide which drugs should we be given and things like that. This is an example of a project that was done at the Genome Center, just illustrating this, but the power of sequencing to really understand disease in a way that we didn't know about before. So this is a project led by Nada Javado at McGill, where she basically, again, had lots of patients with tumors, pediatric tumors, glioblastomas in this case, where the underlying cause of the disease, how can what exactly is going on in these tumors was not known. And by sequencing those tumors, they were able to identify recurrent mutations in specific genes and specific pathways. And now it really is changing how they make decisions about which drug maybe this patient should be getting or that patient. And again, this is an example of something done in research that is becoming more and more done in a clinical setting now. So those are examples of application at the Genome Center. It's not my research per se. My research is much more around epigenomics, if you're familiar with that. just a quick overview. I'll get back to that a bit further down in the presentation in terms of what we do specifically here. But just setting the stage for what I'll talk about later. So epigenomic is really, there's multiple definitions. My definition is really characterizing the genome and the epigenome of the cells. If you look at all of this tissue diversity in the human body, each of these cells has the same genome, but clearly it's using that genome in different ways. So in different tissues, you'll have different genes that are expressed. You'll have different enhancers, different promoters. So different parts of the genome are activated and are used in different ways in these different cells. This is true in development, but this is also true in disease. So characterizing again, which genes are expressed, the methylation of these cells, to understand the underlying rules, basically, of how specific genes get turned on, how. So that's, I guess, the interest in my lab is really to understand gene regulation. How does the genome know which part to activate itself in these different contexts? So again, I'll get back to that a little bit later as well. But all that to say is that the same sequencing technologies can be used to profile DNA, but also to profile the RNA and the methylation state of these cells and so on. So my background is applied mathematics and computer science. I work more downstream of these technologies, looking at some of the computational challenges associated with the analysis of these data. So this slide is already a bit old. It seems like it's always a bit old, but I don't think we've changed our sequencing capacity that much since then. But this is just showing the growth, and I'm sure you've seen, if you've read anything about genomics, I'm sure you've seen this or something similar to this before. But this is really our capacity for sequencing here at the Genome Center. You notice that on this slide, I actually arrived in 2010, so it's like right before good timing. So the burst is, has nothing to do with me, has to do with the fact that we started purchasing these next generation sequencing instruments. But you see the huge jump in terms of capacity that comes from improvement to the sequencing instrument. So we're now at a stage where we can sequence 20,000 human genome at the center per year. And that's really what allows the applications that I briefly described. The fact that the cost of sequencing these genome is now feasible means that we can really do it in a research context, but also in a more applied context. But just underneath this growth of capacity is really this jump in the capacity, I guess, of the sequencers themselves, right? So just this is one of the main Illumina sequencer platform, and you look in 10 years, where did the improvement came from? So I mentioned that typically the reads that we have are very short. So especially 10 years ago, these instruments could only sequence 36 base pair at a time, but they could do that at least in parallel, so 20 million bases at a time, times 8 lanes in this case. So you could sequence with one of these instruments the equivalent of the length of one human genome, but that was not enough to be able to say that you've sequenced a human genome. So the big jump 10 years later is, both the length. So now at least we can sequence slightly longer DNA fragments, 2 times 150 base pair. You see that the density of reads, because this is all being read in parallel using various imaging strategy. So you see that the density of reads that you can read has increased quite a bit. And so that really is what leads to this big jump in capacity that we have. So just again, to put in context, this is the main sequencing technology that we currently use, but it's still restricted to just a few hundred base pair. These other technologies that are the upcoming technologies don't have the same throughput, yet, but they do have this very significant increase in the length of DNA fragments that such that it's more like 100,000 base pair as opposed to 100 base pair. So back to the data associated with that. So again, if we look at one of these sequencers, it generates these intensity files that are coming from really high resolution images. These run over a few days, but you see the size of the data that is initially generated that then needs to be processed into DNA sequences, including qualities. If you have a few of these machines that again take a few days to run, but this already starts to add up quite a bit in terms of total size of data that's generated. And you see the accumulation of these. So you need to process the data, but then you've generated all of these read files, this DNA information, and you'll need to analyze it downstream as well. So you're not quite done, but you are accumulating a lot of data that you need to store, and you want to be doing additional analysis on all of that data, but it's growing very rapidly. To put that in the context of maybe a realistic big project, but still realistic project, we're talking about now, for instance, there's a project that's funded by for which the Terry Fox Research Institute, which you've probably heard about, has received a lot of money to sequence, hopefully, tumors across the country systematically. They're planning to sequence 15,000 tumors. So you see that, so this is a realistic small version of that, but just comparing 1,000 tumors to 1,000 matched control, if you'd be interested in identifying mutations in these tumors that differ from the matched control, that means comparing 250 terabytes of raw data so it's not something that you do on without any tools or without any significant infrastructure. So this is where, so I mentioned in the beginning that there's really 2 Genome Canada platforms at the Genome Center. There's a platform on the sequencing side, on data generation, but I'm actually running a center for bioinformatic analysis and sort of downstream analysis for these data sets. So this is a joint project with Toronto, University of Toronto, Michael Bruno there. So the idea there is that we're developing tools to analyze these data sets, which we also offer to support the various projects. And we implement software in partnership with Compute Canada and Calcul Quebec. So I'll talk a bit more about that. But the types of things that we support, so we support whole genome sequencing, exome sequencing, but also some of these other types of genomic analysis. Again, in some cases, people have the expertise to do that themselves, and that's great. But when they don't, then we can actually provide some additional help for those different types of analysis. We also do quite a bit of training activities, and this is training activities in the context of bioinformatics training activities, bioinformatics analysis. At the end, I'll come back in the context of the McGill Computational Medicine Initiative. I'll talk about other training activities as well that might be more relevant or interesting, but that was just to say that we do some training in these tools. Oops. So I mentioned briefly, I mentioned briefly that we work very closely with Compute Canada and Calcul Quebec, and many of the committees you mentioned that I'm part of is because I'm quite involved with thinking about the compute infrastructure, both here in Quebec and in Canada at some level, to support our own research, but in some case other types of research. But it's just that because we had so much data, in the first instance, we actually set up a compute center at the Genome Center, but that was not enough. So we've really been working very closely with Calcul Quebec to try to adapt and migrate our tools to their infrastructure. So, in some case, we made sort of more easily accessible portal to facilitate for people that don't necessarily have the expertise to run or install all of these tools themselves. But just again, in the context of for people that are not necessarily used to manipulate these types of data sets, we've been trying to set up various tools to support those projects. We've also been developing pipelines for these various analyses that I mentioned. I should say that in all of this, we haven't developed so many completely novel pipelines. A lot of this is really just testing various tools that other people have developed and making sure that those tools can run for the applications that we have and on the clusters that are available. So these are the pipelines that we use, but at the same time we make them available to anybody who's interested because they're already installed on these shared resources. Galaxy is just another of these tools that's more sort of friendly and we use actually this for testing. So this is another tool for bioinformatics analysis. We use it for training and for other things, but again, if you're not so familiar with bioinformatics pipeline, it sometimes can be a good starting point. So I mentioned I would come back to epigenomics, so now I'll talk a bit more about the type of epigenomic data analysis that we do in my lab. So I mentioned that we're interested in characterizing the epigenome of these different cell types to try to understand how the genome works, basically, because we know where the genes are in the genome, but we're still just mapping basically and identifying which parts of the human genome are used to control the expression and transcription in these different cell types. So this is obviously not something that we started. This image is from the NIH Roadmap Consortium. Before that, there was the Encode Consortium. So these are all large international efforts, a bit like the Human Genome Consortium, where we wanted to have a reference human genome. We want to build a reference of what the active state of all of these cell type is, such that we can then better understand the disease state and so on. So this is just another illustration of this. We're quite good at identifying the genes in the genome, but we're still discovering and identifying new regulatory elements in the genome and trying to understand how all of this complex regulation works. And one useful resource to make progress in this is really to generate as many of the data sets as we can that highlight these different elements in different contexts, and then hopefully use that to better understand the underlying rules. So this is an example. This is a bit hard to see, but this is an example, a zoom-in version on the human genome somewhere. So at the very top here, you have 2 genes, and every row in this plot just shows you just a single assay that's been done across all sorts of different cell type and different condition. So you see that you have so in this particular assay measures open chromatin. So this particular assay shows that across almost all of these cell type, this particular position in the human genome is accessible, while there's other regions that are clearly more cell type specific that just show up as being accessible in that particular cell type. So again, as part of these consortium, what we're trying to do is generate these data sets such that we can then use that to learn and try to understand the features, basically understand the features associated with these common these common elements or these cell type specific elements. And one of the reasons we want to do that, number one, we want to understand better transcription regulation, but we also want to maybe better understand when we see a mutation in a gene, we're pretty good at predicting what it's doing. But if we see a mutation somewhere else in the genome, until we understand again these maps, it's hard to know. But it's likely that a mutation in this element is more damaging potentially than a mutation that's hitting in the middle of nowhere. So again, at some level, we're collecting the information to then learn new models on that. So I mentioned the fact that this type of work started really with the Encode Consortium in the US. This was followed by another US consortium, the Roadmap. And we're part of this international consortium now called IHEC that continues this effort, because there's lots of different cell types, lots of different tissues, and lots of different assays that we want to do, and we're trying to coordinate that across the different partners. So you see that Encode and Roadmap are part of this international consortium. We're funded through CIHR. So CIHR had a big initiative to be part of this consortium, and they identify 2 mapping centers. So we're one of these mapping centers, and there's another mapping center in BC. And the effort of the consortium is to try to coordinate these efforts such that we have this library of what's happening in different cell type, especially in normal tissues, that we can use as the basis of other studies after that. So the objective that's written down there is to have 1,000 reference epigenomes characterizing the normal state of all sorts of different cells in the human body. I should say that one tricky part of this is actually obtaining the cells themselves, because if you're asking somebody for brain cells, then they don't necessarily will contribute. So some cells are easy to access. Blood cell types have been profiled quite a bit, but one of the raison d'être of this consortium is actually, you need to work with clinicians doing specific operations to get access to muscle tissues, for instance, and then use those to do the profiling. So that's one of the things that the consortium ends up doing as well. But then we tried to make it such that the assays that are done and the informatics that's done on those is standardized, such that we can compare the data sets and use them. So I'm going to shift gear a little bit. I'm still going to talk about this project, but I'm going to shift gear a little bit now to talk about, because again, I'm more, although we are generating these data, my expertise is more on the data side. So how do we manage the data for this consortium? And so I'll shift gear a little bit to talk about that. So the consortium, just like the Human Reference Genome Consortium, the idea is that we want to generate this data to then be a resource that we can distribute this data to everybody. But in the genomics world, and it also applies to the epigenomics world, even though that's really the core mission, is to make that data available, it's quite a challenge to do that, because genomic data and epigenomic data is a bit like a fingerprint. And so we cannot just release that data, even if we want to, without any kind of control. So there's a big effort and emphasis to make the data available, but in a controlled way, such that we're not affecting the privacy of individuals that are participating in this. So as part of the consortium, we tried to make the data available, but we split the data into controlled access data, so data that is potentially identifiable. So again, we limit the access to this. People can still access, but they need to apply to request access to that data. And then we also want to have, if it's not identifiable, if it's not problematic, then we want to distribute that data more broadly. So the way it's split, and again, this is not only within our consortium, this is true for other genomics consortium, but the raw data itself, number one, the files are very big, but it's also identifiable as a fingerprint, so genetic information is identifiable. We also have, in some case, clinical and sensitive information associated with these sample. That's also something that we don't want to be releasing too broadly. But then what is fully public are sort of the merged data, what's been processed such that there's no longer identifiable data, and some basic information about the sample that again would not reveal the identity of that data. Because again, these samples are coming from real individual, in many cases that have just gone through some surgeries or something like that, but they're not necessarily, we wouldn't want those data sets to be identifiable. And we try to make it, again, the problem is not that we don't want to share the data. The question is really, how can we share the data without making it reveal the identity of these participants? So this is one of the things that we ended up doing for the consortium. So we came up with a strategy to build the data portal to manage the data that's generated as part of the consortium. So we have these different groups in different countries that are generating data, and these data sets are submitted to these international archive that controlled access to this type of sensitive data. But we felt like this was not enough to make the data accessible through these access committees. We also wanted to then create some version of the processed data that could be shared and could be made available through a portal for the data. So this is really what we've been doing, is really build a portal for the processed data and actually coordinate this overall effort. So this is work done by David Bugeaud in my group. So we actually initially were building something just for the Canadian groups, because there was 2 groups in Canada, and so we were setting this up for the Canadian group, but it ended up being adopted by the consortium as a whole. So now we're collecting data from all of these different groups, and we're making all of this epigenomic data sort of as available as possible to the various groups. So I'm describing some of these projects sort of really in a historical way, but since that project, actually, we've been continuing in that direction quite a bit of working on how to make the genomic data sets more available, and taking into account the sensitivity of these data sets, but also making it useful for research. So just continuing on epigenomics data for a bit. So I talked about the IHEC data. So now we're trying to take all of the data that we've generated as part of the consortium. Initially, it was just done by the different projects separately. So now we're trying to reprocess everything and make it available to the community. So I'll go back for a second to this slide. So in the current state of the consortium, we had independent groups doing all of the processing separately and just submitting the data together. But what we'd like to do now is reanalyze all of this data such that we can make a more unified resource available. And this is where it gets fun. So you'll see, so again, to be honest, we really didn't anticipate the complexity of doing that, I think, as a consortium. So our objective, I think, is noble. So this is all data that we've generated as a consortium, and now we're trying to reprocess it and make it available into an even more useful resource. But now, in order for us to do that, we actually have to go through the same process. We ourselves have to obtain access to the data sets that have been generated by the other groups. Each group had their own access committee. So we need to go through this step of asking the 7 official groups, can we access your raw data? So that was first step. That took us almost a year. Second step, we need to download the data ourselves, because we've all submitted the data independently to the same archive, but we need to download it. That's another year, actually. And we're not done yet, actually. So we're still downloading the data that we ourselves have generated as part of this. And now we have to, we've done our best to try to annotate the data we're struggling with that as well. So we realized that even in sort of, as we had agreed with some of the things that all of the data sets should share, that turns out to be, we weren't precise enough in certain aspects such that there's differences between the differences. Another close to a year. I mean, I don't remember the specific dates, but we've been doing this now for, it must be 3 years that we've been trying to do this, to be honest. And we're just at this level of trying to reanalyze the data. And the heavy use of resources compared to everything else, by now we're like, this is actually a piece of cake, because we've been just slowed down by everything else. So this is a slide from Jan Joly, who's also working at the Genomics and Ethics group at the Genome Center. Within IHEC, because this was a grouping of 7 different countries with different projects, we had slightly different rules as part of our data access committee. So you look, and there's not almost no rule that's the same for everybody except acknowledgements, right? Everything else in terms of IP, in terms of, there was some small differences throughout the giant consortium, such that it makes it very difficult, again, to ourselves. That means we have to go through the process, follow different rules. Again, it's been a bit of a challenge. I've talked about this quite a bit already. Downloading the data can be a long endeavor. We need space anyway. So it's really been very, even though it started with good intention, through this consortium, we really realized how complicated this was. And so that was like the scary part of my talk. Now we go back into hopefully optimistic, things will be better in the future. So I don't know if you guys have heard about the Global Alliance for Genomics and Health, but this is a project that, sort of also, we're not the only ones who've realized that this is a major challenge. It's a challenge in our Epigenomics consortium, but it's also a huge challenge for the genomics world. If you look, so this is again already dated by a few years, there's thousands and thousands of these data sets that are now being generated, but they're all being collected, hopefully put into these archives, but there's lots of limitation in terms of how we can access these data sets. If you project into the future a little bit, there's going to be millions of these genomic data sets. So clearly the model that I've just described is not going to work, because, we can't do it with 1,000, we won't be able to do it with 10 million. So this alliance is looking at ways to change the way we do things, basically, and make these data sets more easily accessible. So it's an alliance that groups people from all over, and specifically for genomic data, I should say. But this is an alliance from lots and lots of different members. Again, lots of people that have been involved in these types of projects and realize that it's not going to scale. It's interesting the way they're set up. So they're set up in terms of technical streams. So these are sort of problems to solve, like what kind of cloud technologies are we going to use, or what kind of tools are we going to use for genomics, and so on. So they've got these technical problems that need to be solved. So we've sort of worked as a consortium to identify there's data security and identity management and these types of things. So we identify some of the challenges that we all face. And then on the other axis are these real world projects that have faced some of these things and are going to try to contribute to solve them. So this is the structure of the alliance. And then you have tons of projects that are part of this alliance that are used as these test case of how can we do this. And so one of these projects is one of our projects. So we're participating in these discussions of like, how can we do this better, basically. And so I won't go too much, because I guess still have a bit of time, but I thought because people talk about open science quite a bit, and I thought it's helpful to think a little bit about some of the technical challenges that are associated with that. There's some social challenges of just getting people to share their data, but even when you want to share it, if there are some of these questions about personalized information and so on, they're really challenging problems to solve in terms of how technically can we do this. So anyway, so prior to joining the Global Alliance, we had written a project to try to do this better within Canada, so it's called CanDig. So we were looking for ways, so just going back to the very beginning of my presentation when I talked about the different Genome Canada centers, we generate genomic data mainly in BC, Toronto, and Quebec. So we have got these 3 large centers. How can we share data for some of our projects together? We cannot put all of our data together because some of the data belongs to us, some of the data belongs to them. And again, there's some sensitivity. So we've been working on this model to actually connect our 3 centers in a way that we could authenticate people and provide some access to the data in a better way. So this is one of our projects that we have on just for sharing genomics data that fits with the objective of the Global Alliance. We have another project that follows on our work from IHEC, where we're trying to do the same for epigenomic data, because I've showed you the pain that we've been going through as a consortium to try to analyze and access this data. So we're trying to build these platforms that will allow better control and better sharing of these data sets. So again, this is just another of these data sharing projects that's now also part of the Global Alliance. So with both of these projects, we're now in discussions with lots of other projects to figure out, how can we build infrastructure to share genomic and epigenomic data. Okay, so this was starting from the Genome Center and expanding in some of these international projects that we're part of. To finish, I'd just like to come back to McGill and some of the activities that we're doing. So this initiative is a new initiative that was initiated within the Faculty of Medicine, but obviously involves other faculties as well in terms of the kinds of things that we'd like to do. But this, again, just realizing how much these new high-throughput technologies are generating data, and how we want to potentially better coordinate some of the activities within the university to do that, such that we have sufficient storage, sufficient compute, and appropriate methods to enable the types of projects that I was talking about. So this is really an initiative just to scope out the work that's happening within the university and see what could be done to further improve this. So the objectives of the initiative is really, how can we, it doesn't fall within one department, this is across department, this is across faculty. So how can we facilitate research in computational medicine? What are the current limitations? What, again, can be done? A big component of the initiative is thinking about training and training, existing training programs, and whether there are additional training that could be done as part of that. And then the last part is on potential, having a way to coordinate partnerships with others. So these were the objectives that were sort of set a couple of years ago. I'll go into a little bit more what we've done to try to achieve some of these objectives. So this is again, just an initial collection of people that were interested in this that we put together and have been trying to think of what could be done to again, improve computational medicine within McGill. So one of the things that we started this year, and I'm quite proud of, to be fair, to be honest, I thought it was quite exciting. We started this because we didn't know what else to do, and we came up with that. But the idea is that we found that there's lots of people that are interested in applying quantitative methods in medicine, but they don't necessarily know where partners are. So this is some kind of a dating for scientists program where we felt like, it's one thing, so we, as part of this initiative, we would meet people from different departments and everybody would tell us they have data, but they need help to analyze it. And I'm like, okay, well, this is not helping us because you need to be a bit more precise in terms of describing the type of data and what you're trying to do, and then we'll see. So we came up with this proposal to say, well, it doesn't need to be complicated, but if you're interested and have data and you would like to find somebody to help you with this data, write us a one page to describe what you have and what you're trying to do. And then we'll try to find people that maybe are interested and can help. Personally, I thought that we would only get requests and that we found nobody who's interested in working on those things. I was very pleasantly surprised where we got tons of application in terms of people describing us what they're trying to do, and then quite a bit of interest of people actually interested in helping because lots of new faculty that were looking for data sets for their project that then, and so we had this as a two-stage process where you would initially just write a one-pager, then you would have some time to find a partner, and then you would submit a joint application. So I don't have the specific numbers, but I think we got 40 or 50 expression of interest, and then we got 20 pairing projects, and we were able to fund about 10 or so. So I think it was sufficiently successful that we were really going to do this again, because I think it is one way of really helping people connect, which I think is one of the challenges that we have actually in this, given that it's this, so spread out in terms of the university. So this is one of the things we did, which we'll do again. Another thing I mentioned briefly, but in terms of training, we heard that there was a lot of need, at some level classes, but also at the level of workshops and just basic skills in programming and data visualization and things like that. And again, there's a lot of activities already, lots of workshops that are happening, but oftentimes people have a hard time finding what's happening or whether there's gaps or not. So we've been trying to more systematically organize now or help or support workshops that are already being organized. And one of the things in particular for this, and again, I think this is something we'll want to repeat, there were a lot, I know for instance in my department, there were a lot of students that were self-organizing workshops for other students, which I think is great. And I was like, how come they have no help in doing that? It's like, so the idea here was just to help them book rooms, get a bit of food, and maybe get a bit of money if they are willing to do all that work themselves. So that's another thing that we've done a bit and which we are hoping to do more. Another problem that we identified as potentially a problem, and I'm not sure we've solved this, and again, I'm putting this out there, this is work in progress, and I'm looking for feedback and input into this. But I know that for me, one of the challenges I had as well was to find courses that could be relevant for some of my students, given that they're working on things that are across departments and across. So we've tried to organize lists of courses that might be relevant if you're working in computational medicine. And again, it's obviously not complete and needs to be updated, but this is another attempt to just basically collect information that's currently very distributed and hard to find from my perspective. The last thing, and this is again another work in progress, but going back to this idea of how do you find people that maybe share some of your interests. We've been working on a sort of a lightweight method that would be sort of fun. This is mostly for professors in this case, but to just find people that would have shared interests. Again, because people working in computational medicine, sometimes there's one or two in one department that's at the Jewish and things like that, and so you don't necessarily run into them. And so this is another attempt which we need to now continue to beef up to actually help people find people that have shared interests. So that's it. So that's what I wanted to talk about. So again, I've talked about all sorts of things. In the context of the last topic, it's really for feedback more than anything else. So if you have suggestions or comments, don't hesitate to ask now or drop me a note. So, but thank you.
Thank you so much. So if you have any questions, I have to give you the microphone. You need to speak into it because we're recording. Okay. So any questions from the audience? I might start just out of curiosity. I've heard of some of these initiatives that you mentioned at the end, like the research match.
Yes.
I thought that one was great. Has anyone here used or any of these initiatives before, submitted an application? No? Really? Research match? Oh, that's surprising. I've seen it go through, and I feel like a lot of people at the MNI at least had submitted some applications.
So we had very good response, and again, we want to convert that into a more regular program. So hopefully.
And I was surprised because I think I hesitated myself to submit some data, and then once I saw the projects that were, had gone through, I was like, oh, I could have totally participated in this. So, yes.
Oh, okay. Well, thank you for your talk. It was very nice. So I have a question about what do you see when going forward to see this, I guess, this tension between privacy and open sharing of data. So in particular, I think this is not just limited to genomic data sets. It could be other, like medical imaging data sets or electronic health record data sets. It does seem a huge challenge. I mean, what?
I think it's a very good question. I think, in some ways, I think genomics has an advantage that there's been a lot of work and a lot of thinking along those lines. I think a lot of these, but it's a difficult problem, right? So even if you convince people to share their data, and even if the data is organized, one advantage I think we have in genomics is that the data itself, at least the sequencers, we almost all use the same sequencers, so there's a bit more homogeneity in terms of the data we're trying to share. So I think that helps, because otherwise you also have the problem of very heterogeneous imaging repositories and things. We don't even have that problem, but even then, it's incredible. Then, we keep adding technical things that we need to solve, even if we bypass that one. I think there's sufficient investment and pressure, though, to really find some of these solutions. And I think at some level, again, we haven't solved heterogeneity and lots of other problems in terms of data acquisition and how you're going to actually have data that's comparable across the different sites, right? So that's, but, in terms of regulation, in terms of agreements, it's really, so, I think one of my concerns I have, and that's also why I wanted to talk about it this way, is that I think people don't seem to quite understand the level of complexity with this, right? And how many technical challenges there are. So when so saying open science is going to happen, it's just, it's really, and it's funny too, because it's a question of definition, right? Because I would argue that in genomics, it is all already open, because it's as open as we can be. We all, for the most part, submit our data to archives, and then the data is accessible. So we're already very far in terms of at least making it available. It's just that then it's not available because of you can't download and getting access is complicated. At least we're past just the fact that the data is not compatible, but we already still have all these other problems. So, I look at the Open Science Initiative at the MNI for things. I haven't been following that very close. I think it's great, but in their case, I think they not only do they have these technical challenges, but they also have more challenge, I think, is figuring out what data is going to be made accessible, because they haven't resolved that for something.
Just to follow up on that question, because this a similar type of privacy versus open data also exists in other types of big data, Google, Facebook, or 23andMe. I'm curious, how do you see? How do they address?
So it's a good question. So I got invited to a weird conference on transportation, and I was like, why are you guys inviting me? But they were saying, we're interested in knowing how you guys in genomics are dealing with large data. And I didn't expect this, but we actually hit privacy was exactly the thing we had in common, because they have the exact same types of questions and concern. So it's funny how much there is in common indeed in terms of some of the challenges that we face in completely different areas. So I would say, yeah, I didn't know why I was going there, but we were talking about the same things. Like one example of something that we talked about was talking about Google, for instance, right? And in the genomics world, we have companies that are trying to use the genetic data, and then we have these more open consortium. And so, and they didn't, I think again, we were ahead of them in thinking about this because, like, again, just to talk about genomics and privacy, you know, it's one thing to give your genetic data to a company like 23andMe. I actually don't want to do that, right? But they're making it very appealing to do that because they're packaging that, but underneath this, they're going to be using that information for their own purpose. That's right. But what I like much better are these more open scientific resources that are being set up where they say, give us your genetic data, we'll give you some version of what 23 is giving, but we'll do that, and the data is actually going to be used for science, not for 23andMe shareholders. I mean, I'd go for that. So I was giving that as an example as the good way to bypass some of these questions that they were struggling with in terms of, do we give our data to we can't access the data to Google. And I was telling them, you know, if you guys set up some mini Google, well, good luck, but I mean, but if you set up, because you can retrieve your data from Google and you can upload it in something else. So that's the strategy that's been happening in genetics, is that because 23andMe, they sponsor the cost of generating the data quite heavily, but then they can't hold you from taking the data. So at least you can take it and put it in a public archive. So.
Thank you.
Questions?
Really quick question, maybe a long explanation, but on slide 2 maybe, what exactly does your raw data look like? I saw something said imaging, I saw sequence,
Yes.
I saw something else. I have no idea what you're looking at. Are you just looking at a bunch of C's and A's and
Yeah,
T's
good
and
question.
G's,
So I didn't
or-
know how much of that intro I should be putting in, but so the basic way these sequencers work, so the ones that we're using in the standard way now, is really imaging based, where so these millions of short DNA fragments are actually attached to a slide, and then DNA just has 2 strands, and you're basically, just like in our cells, you're adding you're copying one strand to make the other strand. Right, so in our that part is clear, I guess, right? Okay, so the different sequencers basically use that underlying principle, but they use different techniques with like probes and like such that basically you have different lights that emitted when you're incorporating these bases. So the underlying data is really just taking images where one base at a time gets inserted, and whether so you got the 4 colors and so on. So the underlying data are these images, then those are converted into just intensities of these different color channel, and that's converted into then these short sequences. So it's ACGTs, yes. I didn't answer it properly.
Somewhat. So, okay, so I got the AGCT.
Yes.
But how is this information kept in sequence? The length of the DNA sequence, or is it on this small peptide, or on this?
Okay. So then there are 100 base pair reads, right? So they're relatively short, 100 base pair CGCT. That information by itself actually could also be compressed a whole lot more, such that it wouldn't take that much space. I mean, they're big numbers, but we can compress that quite a bit. Typically, though, we keep also information about the quality of the call, because, in some case the images are there's 2 fragments that are very close to each other, so you're not sure about the call. So we're also keeping the information about the quality of the bases, and that's what adds up. But typically, these files are really just like millions and millions of lines that are just the sequence ACGT followed by qualities associated with each of those. So that's the underlying raw data. Those can be compressed, and what we use after that, The next big step, so we've got millions of these short fragments. The next big step is mapping onto the reference genome. So the next big set of raw data is really the read itself, so the sequence of ACGT, and where does it go on the human genome. So we then have information about location on the human genome, additional quality about that, whether it's a perfect match, a mismatch, and things like that. So this is another main format that we use. And that one actually is interesting. I mean, just thinking more on the computer science side of things, it's interesting because this one can also be compressed using very interesting methods because, again, you know where it goes. So you can use, once you know where it goes, you don't need to keep as much information from the read. You just keep the positions that are different and things like that. And so, but even then, those files remain quite big unless you get rid of the quality and information from the capture. And that's where we have some wiggle room, I guess, in terms of reducing the size of the data sets. But given that we're still developing the algorithms downstream, people are really resistant to get rid of the information. But that's why these files are quite large. For sure, by the time we, I guess I should say, so I mentioned the raw data, secondary data. The third data type, especially in the genetic world, is we forget everything about the reads and we just keep information about the variants. Which position, based on all of that, once we've mapped it in the reference, most of it is the same as the reference, and then we just keep a file with the information about what's different. And that's tiny in some ways by the time we get to that, well, compared to the rest. By then it's definitely a 1,000-fold reduction in terms of size of the files. But it depends. So it depends where you plug in. There are definitely applications for which the size of the data is not so problematic, but the underlying raw data is very big. It used to be that we would keep the images, but thankfully we don't do that. But that's in part because we can't. So that was an easy choice. I should also add that so the size of the data is a challenge. The processing is also a challenge because, again, but it's highly parallelizable because there are just millions and millions of reads, billions of reads. You can really distribute the jobs. Most of the steps, especially at that level where the size of the data is biggest, that's really highly parallelizable to the point where now it's also finally we're making the migration to using GPUs and specialized hardware for these steps. I don't know about your research, but I mean, we've been talking about this for 10 years. It's taken a long time for genomics to make that transition, but I think it's actually now finally happening for some of it, at least. There's really some steps that have been optimized, which makes sense to be using these. So there's a lot of development there as well in terms of how to do that efficiently. Yeah.
Thanks. So very interesting initiatives. I have a question about the accessibility. So you said that in the public domain, and when you archive it, the public has access to it.
Well,
who exactly?
Yes, that's a good question. So for the public tracks that's not identifiable, this is really open access. Anybody public can access and see it because there's nothing sensitive about it. So this is really completely open. The controlled access, the way you have to make a request, in theory it's anybody, but in practice you have to be a scientist because you need to explain your research question, and then that gets reviewed by a group of scientists and access committee that involves scientists and in some case patients that have contributed their data and so on, that basically look to see what are you planning to do with this data. And also has a few checks. You're not going to use that data to build a database to screen people for insurance purposes, right? There's something like that, obviously. And so there is just this additional check where you have to describe why are you requesting access, what are you going to do with it at a high level to make sure that it's scientifically justified and that it matched what people initially consented when they gave their samples.
So this is just in Canada or anyone in the world?
Anyone in the world. I mean, this is really, well, there's some local projects that are using the same type of approach and system, but what I was talking about here are really these international archive where when you submit your data, you also will have a data access committee. And so anytime somebody requests, from anywhere in the world, requests access to your data, you get ping, and then you get that request to your committee. And they don't release it until you give the okay to that. So, but that's, again, in the genomics world, that's really been the default for projects where this is what people are expected to do. But it's just, it used to work better when the size were smaller, but now we're like, we want to retain the same kind of principle and same idea, but technically, even if I give you access to 100 terabytes in UK, it's going to take you some time to download it. You probably don't have spare 100 terabyte, to either, right? So it's like, so we're just trying to adapt those principles to this new reality that, we can't just download all these data sets and make multiple copies and so on. So but to go back to the question on other data types, I think that it's the same principle that's also at different level. It's also applies to other data type, whether it's imaging and things like that. And so I think that is the right model, but there's a lot of technical things to work out in order to be able to do this then efficiently.
On this, the business of sharing data, is there a concern with your type of data? Is there a concern with people, the people who own the data, not wanting to fully share it because they want to be able to exploit it first and publish papers before they give it away?
Well, there's a bit of that, but again, I think, there's the real people that give the data are the patients, right, or the individuals that are contributing to those studies. So they definitely, I think, have all the rights to their data and to share it in the way that they want, right? So at the level of initial consent, the people contributing their samples, I think, have all of these rights, and then that's what needs to be specified in the initial consent. I think, to me, so the scientists that then are using public money and other people's samples to generate data, I don't think I have a very strong case to say that this data belongs to them. I think in our community, that's pretty well accepted that, at some point this data really needs to be, understandably it needs to be quality controlled and exactly when does it get deposited. But I think in our community, it's quite, people really expect that with a genomics paper, that genomic data set, there's a requirement that it's accessible to one of these archives. And if it's not, there's exceptions, but that's really the expectation. So I think that's just an advantage, again, in our community. And that really comes from the fact that it started like that with the Human Genome Project. And all of these projects downstream are sort of the same people that started that and set it up that way. So I think it's, I don't want to say it's perfect, but it's not so bad in terms of how it works, and it's definitely the expectation.
Another small question. On a couple of times on your slides, you were talking about the amount of data. You said at 30x.
Yeah. Oh, yes. So this I didn't explain at all, so it's a very good question. So when we sequence a human genome, all we have are these short reads that we map onto the reference. If a particular position of the human genome is just covered with one read, we won't be able to tell whether there's a mistake in the sequence, or because our goal is to see whether the genome we're sequencing is different from the reference. So we need 30x on average. So we need to cover the genome with multiple reads at every position to have confidence in what we see in the read. So, because the reads are not perfect, we may, there's 0.1% errors when we sequence the DNA. So we want multiple pieces of evidence, and that comes from having, so typically we request 30x because, there's 2 chromosomes, and then that means 15x on average at every position for both chromosomes. such that we have confidence that if there is a difference, we'll have multiple reads telling us that there's a difference and we'll be able to call it. So a very good question. I mean 30x, sometimes it's a bit less, sometimes it's a bit more, but the main idea is that we need, in order to sequence a human genome with confidence, a new human genome with confidence, we need to sequence it multiple times, basically. So it's some kind of, it's also not perfectly even, right? So if it's 30x on average, it means some parts of the genome will be at 10x and other part will be at 60x because we randomly sequence the genome, if that makes sense.
Does this X factor, when you're doing these much longer chunks now,
yes,
is that number going to go down? You need fewer?
Excellent question. Absolutely, yes. Because one of the reasons why we need multiple reads is because we also don't necessarily know exactly where they go in the genome. So the genome is repetitive. So some parts of the genome with 100 base pair, we don't know if it goes there or if it goes there. So one of the reasons to ask for 30x is to have this increased confidence that we're placing the reads correctly. But when we have a 100 kb read, that confidence has gone up already quite a bit. So with these longer reads, which are more expensive, we definitely don't target the same coverage. We can do 5x or 10x or something like that. And typically, right now, if we have to see, this is something we're working on, what's the best way of sequencing a human genome? It's a combination of a few of these long reads, maybe 5x, and many of the short reads, which are cheap and so we do a combination of short reads and long reads to have high confidence human genome. Good question.
There have been a couple of reports in the news in the last few months. There was a murderer who was caught and convicted
Yes.
out in California or something.
Yes.
So is it truly possible to completely de-identify a particular person's genome?
Well, good question. Also scary. I mean, like one of the things with DNA, and in this case, one of the challenge with this is that your DNA runs in your family, right? So it's not just what you decide; it's what other people in your family decide in some ways. So a lot of these things. So not only is it hard to de-identify, it's also, even if you yourself never contribute and don't want, you can be caught because your cousin submitted their data to this database, which is something that, going back, we're talking a bit about consent. It's a bit weird how consent works because, what does it mean that, so, if my sister consents, it affects what I do, right? Or what it means for me. So, I mean, it's really quite, to me, just to answer the question, to me, it is impossible to completely de-identify, and there should be less emphasis on that and much more emphasis on regulation of what people do with this information. And so, and again, we have a big group that works on these types of questions at the Genome Center, and I really enjoy these conversations with them because it's, sometimes they're like, one time at our journal club, one of the people from the ethics groups started his presentation by saying, sequencing the human genome was a step backwards. And I'm like, wait a minute, step backwards. It's like, because they're putting the emphasis on, they were going to talk about privacy and the dangers of this, but it's like, cost benefits. There's, this is slowing down science that actually is helping a lot of people. And if you think about the information that is in your genome versus the information that people are posting on Facebook and Google, and they're doing all these things that actually are much more damaging to their privacy with no questions asked, and then they're freaking out about their genetics, which is also useful. But there's something special about genetics because it runs in the family, because it's permanent. But it's also a right balance between cost benefits from my perspective, right? So.
I'm not so sure about that. We will see where it all ends up.
But for sure, one thing to me is that, the way this information is used should be tightly controlled by law, which is not always the case in different jurisdictions. That's the part that I don't understand, because these people are really sort of watching what the scientists are doing, which is good, but they should also be putting pressure on on governments to really regulate the way this information and other private information is used much more, from my perspective. All right, thank you.